Hi everyone, so today I'm going to explain you about the structure of DNA. Of DNA, as you can see, DNA is a double helical structure. It is a double helical structure. So say the double stranded DNA, these two strands will intertwine to form a double helical structure DNA. This is the internal chemical structure of the DNA. Each strand is a polynucleotide, that is, each unit is made up of many nucleotides, that's why it is known as a polynucleotide. So see, each nucleotide will consist of three groups, that is, the first one is phosphate group, and then sugar group, and then base group. This base group is nothing but these are the nitrogenous bases. Four nitrogenous bases are adenine, guanine, thymine, and cytosine. The sugar here is called as a deoxyribose sugar. Why? Because it lacks hydroxyl group at the second position. Because of this, the nucleotides in DNA is called as deoxynucleotides. Nucleotides are bonded by bonds known as phosphodiester bonds. The nucleotide of one phosphatide binds with the oxygen of the neighboring nucleotide. Thus the sugars and the phosphate groups makes up the DNA backbone. 5-carbon sugar on the left and the 3-carbon sugar on the right. The top strand orientation is 5-3- and then the down strand orientation is 3/5. Like this, the orientation will be there, which is also proposed by Watson and Crick. That's why it is also known as Watson and Crick model. Each base forms multiple hydrogen bonds with the complementary bases which are present in the opposite sides. Thus each unit is known as a base pair. Thymine pairs with adenine with the 2 hydrogen bonds. Cytosine pairs with the guanine with the 3 hydrogen bonds. The thymine and the cytosine are known as pyrimidines, which are characterized by their single ring structure. So adenine and guanine are called as purines, which are having double ring structure. So the geometry of these base pairs is the same in the helix structure. Geometry is different. Pi interactions will be formed at the bases stacks, and then they will share the electrons. That is, the electron probability sharing will be there at that point. Regularity of this alternating structure will form into two grooves. They are defined as major groove and a minor groove. They are the spaces. Grooves acts as a base pair recognition and then binding sites for the proteins. The major groove contains the base pair specific information. The minor groove contains largely base pair non-specific information. This grooves pattern is because of the hydrogen bond acceptors and donors that the proteins can interact with the grooves. This way the DNA will act like DNA sequence specific or non-sequence specific manner. It allows the proteins to place successfully in the genome to carry out their designated tasks. The double helix DNA structure.